Hello viewers, I am Anil Kumar Jain from Jaipur, India. My topic of lecture is genetic engineering, cloning and karma. So why this topic is important to us? Let us first understand that. In the second half of the last century, a lot of progress has been made in the field of biology. Genetic engineering and cloning are the two most important research work. In it, now possible to create organisms with desired characters, remove some unwanted characters, rectify their hereditary diseases, and make their carbon copy, means same type of shape, size, and color, etc. These developments put religious teachers into doldrums because when creation of an organism and its physical development is in our hands, then what is the role of karma? Whether religious beliefs are wrong now, we will discuss here these points and try to understand them in the light of Jain karma theory. So my lecture will be in two parts. First I will take genetic engineering and Jain karma theory. Let us first understand what is the building block of an organism. Cell is the basic unit of all living being. All organisms are made up of one or more cells. Human body contains approximately 10 trillion cells. Nucleus of each cell consists of 46 chromosomes. DNA are organized in chromosomes which contain genes. So here I tried to show in the picture, genetic engineering is done at gene level before zygote is formed. New life starts at zygote level. This we have to understand here. So I have shown here in the picture, this is the cell. The nucleus is here. This nucleus is having chromosomes inside it, and these chromosomes are having DNA, and this DNA is consisting of genes. So segment of the DNA is gene. So as I was telling that new life starts at zygote state, so I have shown here in the second picture that sperm are uniting with the cell, they are forming the zygote. So new life starts here at zygote level. What genetic engineering is done before that in this nucleus or in this sperm cell. So in genetic engineering, what we do, genetic engineering can develop the desired character. It can manipulate genetic information artificially. DNA of an organism may be altered by changing its gene. Gene of one organism can be transferred into another DNA. So I have shown here in the right hand side picture, it is shown this is the part of DNA, means it is the gene, and in this gene some part of it is taken out, or we have to implement after removing this. If we want to introduce something here, we can introduce. So human gene can be introduced in the bacterium. Bacterium genes can be introduced in the plants. Human genes can be introduced in sheep, etc. And fish genes can be implemented in these plants. So following things can be done through genetic engineering. We can destroy or inactive a particular gene. We can delete a particular gene, replace or substitute the extracted gene with the gene of some other organism, or insert or add a gene of another animal along with these existing genes. So what is the process? A gene of interest is taken out from the cell the gene is inserted into the DNA of another host cell. The host cell now contains recombinant DNA. The host cell multiplies after that. So I have shown this in the picture. Thus these are the part of gene of DNA 1, and this is the part gene of DNA 2. So suppose I want to cut this gene from here, so it is like this, and suppose I want to introduce the gene of DNA 2 into this. So what will be done? Something is cut from here that I don't want, and something I have taken that I want to introduce into this, and coupled both of them to make a new gene. This gene is inserted into the chromosome. So here another example is given. This is one human cell and this is bacterium cell. From human cell a gene is taken out, and in bacterium cells one part of that is also taken out. So this is shown, thus it is the DNA, and from this DNA something is cut from here like this, and this gene is introduced into this. So we got this new, this is called recombinant DNA, and that is introduced into the genetically modified organism. So this is the process by which we can introduce a gene of one organism into another. What are the benefits of this? High yielding crops can be developed. They can fight various harmful insects and pests. Longevity may be increased. In case of E. coli, scientists found a gene. If that is immune, its longevity may be increased. Hereditary diseases can be controlled by genetic engineering, because we know which gene is creating the problem. Hereditary disease is created by some genes, and if we take out that gene, the hereditary disease can be controlled and desired characters can be developed. These are the benefits. Now question raised are like this. General belief is that karma defines our future. Genetic engineering has proved that these things are not in control of karmas, they are in our control. Then what is the sanctity of karma theory? That tells our functions of life is governed by our own karmas. When a new species can be produced, then what is the authenticity of gati, jati, yoni as described in religious text? To clarify these points, we shall discuss something about Jain karma theory. As I told you in my previous slide, that new life starts at zygote level, while changes are done at gene level. When it was in the single cell, so role of karma of new species starts at zygote level, not before that. So genes are part of our audaric sharir, means physical body. Changes can be made in the physical bodies. As we know, operations are done to cure various diseases in our body. In the same way, gene can be changed at the microscopic cell level. Fruition of karmas depend upon external causes. This is very important, karma theory. That is, place, time, present feelings. They affect the results of our karma. Karmas can also be changed by utkarshan, apkarshan and sankramaran. So these are our principles, Jain karma theory principles. So there is no problem if a new species is created. Hybrid mangoes and other fruits and vegetables are produced by farmers since centuries. Donkey and horse together make a mule in a natural way. There is a detailed discussion about 84 lakh yonis and 199.5 trillion kull. Whatever new species is created by scientists is within these numbers. Scientists are also assisting in making yoni that we have not seen earlier. The use of genetic engineering, what are the uses? Let us see. Genetic engineering is useful in treatment of diseases like cancer and other, and in the preparation of various drugs and vaccines. Useful in developing high yielding plants, variety of seeds, livestock animals. White revolution to increase milk production, and green revolution for the development of high yielding crops is the result of this. We have seen that earlier grain production was less, now it is more because of the modified seeds. These modified seeds are basically genetically modified seeds. Recently we have heard the name of a GM cotton, genetically modified cotton seed. It was in the news, and now we are listening the other news about the genetically modified seed of mustard. But what are the problem with this? Let us understand that also. However, these revolutions have created hormone and glucose imbalances in the pupil. Some research says then we have increased the milk production by using hybrid cows, but the milk of these cows have created microbial imbalance in our body. Likewise, we have a good production of grain, but these modified bees and modified seeds of grains, they have created glucose imbalance our body. These are the harmful effect of genetically modified seeds through genetic engineering. If we are creating a new animal, it is not only unethical but against the nature. If we are doing anything against nature, it is sin. Now I am coming to the second part. It is the cloning and gen karma theory. Before coming to the cloning process, let us first understand how the birth takes place. As I have shown you in my earlier slide, that when sperm and ovum is meet, an embryo is formed. When the sperm of a male meets the egg of the female, the embryo is formed. In this embryo, some chromosomes are of male and rest are of the female. The heredity qualities of the male and female, both parents, come in this embryo. As this embryo develops into a child, the heredity qualities of parents begin to appear in its personality, such as height, color, inherited diseases, etc. So an embryo is formed when sperm of a male meets the egg of the female. So life starts with a single cell at the zygote state, that I have clarified earlier also. So is in this stage life starts. So what is happening here? One cell of sperm, another cell is of ovum or egg cell. They are meeting. They are some chromosomes of sperm, some chromosome of egg are mixing, and zygote is formed. So at this stage, new life is coming. So what happens in cloning? So cloning is a process of producing individual organism with identical DNA by artificial means. In lower organism, cloning happens all the time in nature. Some organisms produce clones through asexual reproduction. In the field of biotechnology, Cloning is the process of creating genetically identical copy of a cell or an organism. So in this process, nucleus of an egg cell of the female is removed. This nucleus-less cell is kept in a safe place. Another cell is taken from the skin of the organism called donor that is to be cloned, maybe male or female, and its nucleus is carefully separated. This nucleus is implanted in the nucleus-less cell of the female. Nucleus-less cell means enucleated cell. This new cell doubles to develop into an embryo that is implanted in the uterus of the female, where it develops normally. Chromosomes of the newborn are the same as those of the donor parents, and its appearance is exactly like that of the donor parent. So I have shown this in the picture, that this is the egg cell of the female. Its nucleus is removed by some technique, and now whatever we are getting is the nucleus without nucleus, the cell. It is called denucleated cell. So suppose we want to create the identical body or identical organism of another living organism. So we will take a cell of that organism, and here also we remove the nucleus. This nucleus is also kept in the safe place, and this cell without nucleus and this nucleus are again mixed, fused together. So they are now having a cell which is having all the characteristics of this new cell, same as what is having here, because DNA in this cell and DNA of this organism which we want to clone have the same identical chromosomes. Then the normal process of birth takes place. So these experiments were carried out for decades, but final success was got by Dr. Gordon in the year 1962, when he successfully taken out the nucleus of the frog cell, and then this without nucleus, the same process he has followed, and now he has taken another frog, and of that frog he has taken the cells, and from these cells he has taken out the nucleus, and that nucleus is introduced, fused in this denucleated cell, and normal way the process of birth has taken place. Another scientist, Wilmert, has created the clone of sheep. It was the year 1996 when he successfully created the clone of the sheep without using another sheep, from the single sheep itself. So what he has done, he has taken the egg cell of the sheep and enucleated, means they separated out the nucleus from this cell, and it is kept here in safe place and without nucleus. And another cell he has taken is from her skin. So here also he has removed the cell and it is without cell, and this nucleus is introduced in this cell which is not having cell now, and both are fused and normal shape has taken place. So the sheep name to this sheep, cloned sheep, was given as Dolly. Eh, So now questions are raised because of this process, because since all the hereditary properties of the clone are similar to the donor parents, so it is a clone. So is the clone a part of donor? This is a question. Since the process of creating an organism, there is no need of male or female both. It means without male, mammals can be created. So is the concept of Garvjanam or birth by womb described in Jainism is wrong. Here, as I have shown you, that both the cells are from the same sheep. In the earlier case here, two different frogs were used, but in case of this Dolly, creation of Dolly, the cells of egg cell of the sheep and uh, skin cell of the same sheep were used. So question is now this, so we don't need male and female both. Yes, that is right. Now, is the cloned sheep is Samurchan living being? Somebody has asked this question many times. People ask this type of questions. So Samurchan means spontaneous birth. Can human be created even in laboratory? According to Jainism, the body of a living being is created by maturation of Angopang Nam Karma. But in this process, we can create a body shape as per our choice. So, what will be the use of Nam Karma? Answer to these questions are like this. Identity of clone is completely different from the donor parent. Its physique will be same, but its existence, life, feelings, happiness and sorrow would be completely different from its donor. Clones are just like twins. We can consider twin boys having same physique but different behavior. Their behavior depends upon environment, how they were developed, how they are getting parvarish of all both the child. Cloning is not possible with a single cell. It involves two cells, one egg cell and other some other cell. So new life comes in zygote cell. So zygote is formed by two cells, not by single cell. This is very important. So both cells together form zygote. Then the baby grows in the uterus. Rest is similar to the normal process. So it is Garvjan. If we say it is Samurchan birth, then one more question will arise. Can a Samurchan dolly give birth to a Garbhaj? Because Dolly later on, when Dolly became young, it also became pregnant and gave a birth to a child, baby, the sheep. So she behaves like the normal. So question is that whether this is Samurchan or not. No, it is not Samurchan, because Samurchan can't create Garbhas living being. New life is created when nucleus is inserted in the nucleus less cell of female. So cloning is possible without male, that is right, but in that case baby will be only female only, because we are using the two cells of the female only. So whosoever is giving the nucleus, who is the donor of the nucleus, we will get the same thing. If donor of the nucleus is female, we will get female. If donor of nucleus is male, we will get male. The same type of male, physique wise. So it is possible to create baby in test tube, but in that case also two cells are required. This is very important. So what is the role of karma, nam karma? One question is when we can create baby with desired physique, then what is the sanctity of nam karma that are supposed to be responsible for the physique? Answer to this question lies in the karma principle itself. Realization of karma depends upon nimit, means we can say environment, dravya kshetra kal bhav, or we can say place, time and environment. If they are changed, result will be different. Complexion of a baby born in cold places will be fair comparatively to the baby born in the hot places by the same mother. So I think the questions raised were fully answered, but some ethical issues are there. Cloning animal or human is against nature and quite unethical. The cloning process causes problem to the animals because lot of sufferings is giving to them. The cloned human would be unique. Suppose we are creating a clone of a human, it will be unique because it is not a normal practice. So this would take him away from the society and he would be under stress psychologically and socially. So these are some issues. So this is end of my topic. Thank you very much for the listening. Thank you.